So today I'm going to be telling you about my research into the major histocompatibility complex, or MHC. The MHC encodes genes that process antigens and bring them to the cell surface for use in immune recognition. This can be seen in Figure 1. In humans, the MHC genes are mainly encoded onto chromosome 6. However, in zebrafish, a teleost, which is used as a model for human genetics, we have seen that the MHC genes are not encoded onto the same chromosome, and thus we are unable to use zebrafish as a model for the human MHC. Luckily, we can use Holosteans, such as bowfin and gar, to bridge the gap between zebrafish genes and human MHC genes, using the Holostean bridge principle demonstrated in Figure 2. Since the bowfin and gar loci are relatively intact, similarly to humans, they make for a better model for the human MHC than zebrafish. You can see this in Figure 3. Within the MHC genes, PSMB8, the gene that is responsible for cleaving long proteins into peptide fragments, is of particular interest due to its strange nature. There are two independent alleles of PSMB8 that appear to have evolved and be maintained even when one of the two is lost. We divide the PSMB8 alleles into the A or F lineages, and one individual zebrafish genome could encode either an A or F lineage PSMB8 gene. Because we believed that bowfin and gar would make good models for the human MHC, we decided to look into their PSMB8 genes. We were able to find a novel threonine, T, at the 31st amino acid residue in gar, and serine, S, residue here in bowfin. The T allele belongs to the A lineage, and the S allele belongs to the F lineage. These are both novel alleles, and this is the first report of them. You can see them in Figure 4 at the red dots. Since we found the F lineage in bowfin and A lineage in gar, we predicted that each species could have the reciprocal lineage, due to what we know about the other species maintaining two alleles. Therefore, we sampled and sequenced gar and bowfin from across the U.S., seen in Figure 5. Unfortunately, we were unable to find any evidence of the reciprocal lineages, and because of this, we decided to look at available online genomic resources for additional alleles in other species. These other species can be looked at in Figure 6. So far, bowfin and gar have been the only species that contain the S and T alleles. Every other species falls into what we already know from a 2014 paper showing fish genuses and their predicted PSMB8 alleles. You can look at these genuses in Figure 7. So what does this mean? We believe that due to the S and T alleles, those different PSMB8 genes can take the same precursor protein and degrade it differently, changing what is seen on the cell surface. This could have major implications for vaccine development and disease treatment, as a cell could change from being seen as viable to not viable and vice versa. Hopefully, this is something we can study more once we know more about the PSMB8 gene itself. Thank you.